You know, what if I told you that one of the biggest revolutions in modern biology came from a molecule so tiny, scientists almost threw it away thinking it was just junk? Today, we're going to pull back the curtain on a tiny molecule that doesn't build a single thing in your body, but somehow it controls almost everything.
So to really get why this little molecule was such a bombshell, we've got to rewind a bit. We have to go back to this huge, decades-old puzzle that was sitting right at the heart of biology. Okay, think about it for a second. Every single cell in your body, whether it's in your brain or in your big toe, has the exact same genetic blueprint, the same DNA. So how? How does one cell know to become a neuron that fires off electrical signals while another one becomes a muscle cell that knows how to contract? What's telling them what to do? For the longest time, the answer seemed to follow this really straightforward rule, something biologists call the central dogma. You've got DNA, which holds the master instructions. Those instructions get copied into a temporary message called messenger RNA, or mRNA for short, and then that message is used to build a protein, which goes off and does all the actual work. Simple, right?
Well, not quite. See, that simple flow of information didn't explain control. It didn't explain how genes get turned on or off, or maybe more importantly, how they get turned up or down. Scientists knew there had to be something else, a whole missing layer of regulation. They were on the hunt for biology's dimmer switch.
And you know where the first clue to finding that switch came from? Not from some big, complex human study, but from a tiny, transparent little roundworm.
Okay, so picture this. It's the early 1990s. Researchers are studying this little worm, C. elegans, and they're trying to figure out a really specific mystery. How do its cells know when to do certain things as it grows up? They ended up zeroing in on one particular gene they thought was the key, a gene called Lin4.
And this is where things got really weird. Everybody expected the Lin4 gene to make a protein, just like the central dogma said it should. But it didn't. Instead, it produced this super tiny piece of RNA that didn't seem to code for anything at all. It was so small it could have just been written off as, biological noise.
But it wasn't noise. Turns out, this tiny little RNA had a kind of superpower. It could go out, find, and then stick to a specific messenger RNA molecule, the blueprint. And by grabbing onto it, it physically blocked that blueprint from ever being used to make a protein. It was a regulator. It was the dimmer switch.
This was a completely new way of controlling genes. So let's take a second and break down exactly what this thing is and how this incredible dimmer switch actually works. So scientists gave this new type of molecule a name, microRNA, or mRNA. And its whole job is basically defined by what it doesn't do. It doesn't make proteins. Its entire purpose is to be a manager, controlling which other genes get turned on or off. That's a process we call gene expression.
Probably the best way to think about it is like this. If messenger RNA is the architectural blueprint for a building, then microRNA is the site manager who walks around and decides which blueprints the construction crew actually gets to use today. And the process itself is just beautiful in its simplicity. First, a gene's instructions get copied into that messenger RNA blueprint. Then, the tiny microRNA manager finds its specific matching blueprint and just latches on. And boom, just like that, production is blocked. The gene's output is effectively dimmed down through a process that we now call RNA interference. Now, for a little while, this whole thing was just seen as a weird quirk of worms. But then researchers found something that just blew the entire field wide open. Right around the year 2000, the story totally exploded. Scientists found another microRNA, this one called Let7. But here's the kicker. It wasn't just in worms. They found it in flies, in fish, and yup, in us, in humans. This wasn't some weird worm thing. It was a fundamental principle of life, something that evolution had kept around for millions of years. This discovery was so profound it helped lead to a Nobel Prize in 2006. And since then, the floodgates have just completely opened. Scientists went from knowing about one or two of these tiny molecules to discovering that the human genome has over 1,000 different microRNAs, each one acting as a tiny manager controlling these incredibly complex networks of genes. So, why does any of this matter to you and me? Well, because when you have over 1,000 tiny managers running around your cells, if they stop doing their job correctly, things can go very, very wrong.
And this is the really crucial part. Faulty microRNA regulation is now linked to a just staggering number of human diseases. Take cancer, for instance. Sometimes microRNAs that are supposed to keep tumors in check get silenced. while others that actually help tumors grow get cranked into overdrive. And we see similar problems in everything from heart disease to neurological disorders.
But the flip side of that is a huge opportunity. Because we now understand these tiny regulators, we've opened up completely new ways to think about medicine. Scientists are actually designing therapies that can target these microRNAs, either by making more of them or by blocking the bad ones to try and bring our cells back into balance.
So let's just take a step back for a moment and really appreciate why this discovery wasn't just another interesting finding. It was a true scientific revelation, the kind that earns the highest honor in science.
Before all this, we really thought of RNA as just a passive go-between, right? It was a simple messenger carrying notes from the DNA boss over to the protein workers. The discovery of microRNAs proved that RNA itself could be the boss. It revealed this whole hidden layer of control that's really running the show, governing the incredible complexity of life.
That quote just says it all, doesn't it? The entire field of molecular biology was completely reshaped, not by some massive, earth-shattering discovery, but because a few researchers paid really close attention to a tiny, overlooked molecule inside a humble little worm.
And that just leaves us with a fascinating final thought. This whole story proves that even after all these years of research, biology is still holding on to some profound secrets. It really makes you wonder, what else is hiding in plain sight inside our cells, just waiting for the right person to ask the right question? What other tiny revolutions are still waiting to be found?